Okay, good morning and welcome back to our lecture today on nutrition and metabolism. Nutrition and metabolism. And this chapter follows in a chronological order right after the digestive system. So we went over how the digestive system works in the processes of ingesting the food and then digesting it chemically and mechanically and then absorbing the food nutrients and delivering it to the circulatory system, and then finally the circulatory system is going to distribute all of the nutrition to the tissue compartment in order for us to perform the metabolism and the final harvesting of the nutrition in order to get the ATP. So in the end, we want to achieve ATP. So the central point is for us to receive ATP. But this ATP, we need to metabolize carbohydrates. We need to metabolize carbohydrates. We need to metabolize lipids. We need to metabolize lipids. We need to metabolize proteins. We need to metabolize proteins. And we need to metabolize nucleic acids. Nucleic acids. These are the four macromolecules that exist. Pretty sure you guys went over those in biology lab. And you know, when you had these reagents that change color when they detect, remember Lugol would detect the starch carbohydrate and Benedict would detect glucose carbohydrate, as well as the burette. The burette reagent would change color if it is able to detect the proteins. So those are the macromolecules. Those are macro molecules. Those are huge molecules. Those huge molecules, we metabolize them. So we metabolize carbohydrates, metabolism. We metabolize carbohydrates. We metabolize proteins. We metabolize nucleic acids. We metabolize lipids. And then we get the micromolecules. So we have macromolecules that we break down into micromolecules. And then we take the micromolecules and we make ATP out of them. So our terminal target is to get ATP. Why? Because ATP is going to be used by the enzymes. And the enzymes are important for survival, for reproduction. And without enzymes, we're not going to survive, we're not going to reproduce, and enzymes are not going to work unless there is ATP, and we're never going to have ATP unless there is metabolism. Metabolism of who? Of the micronutrients. So we talked about proteins being so big. Proteins are macromolecules. Lipids are macromolecules. Carbohydrates are macromolecules, and nucleic acids are macromolecules. Macro meaning that they are huge. Now the question is, do we get ATP immediately from macromolecules, or do we need to break them down into micromolecules first and then shuttle them into metabolic pathways in order for us to make the ATP? Excellent. So we break down nucleic acids into, from macro is too big, from macromolecules into micromolecules. molecules. What are the micromolecules that we break down nucleic acids into? Excellent. Nucleotides. Nucleotides. Then the nucleotides, there's a nucleotide metabolism pathway that will give us ATP. Are we clear? Then we move to proteins. What do we break down proteins, macromolecules into? Micromolecules, but what do we name them? What do we name the micromolecules that make the proteins? So the peptide is a large part of the protein. Proteins are polypeptides. Those are the amino acids. So the amino acids are the building blocks and the micromolecules that make the proteins. And lipids are made up of glycerol and free fatty acid. And carbohydrates are broken down into first disaccharides, and then monosaccharides. And then we get our ATP. So now do you see what metabolism is? Now this direction is one way. You know, is the highway one way or two ways? It's two ways. This way here, we're going from big to small. We're breaking down. And here we have macromolecules that are being turned into micromolecules. From very big into very small. What do we name that process? If you're breaking down big nucleic acids into small nucleotides, big proteins into small amino acids, big lipids into smaller glycerol and free fatty acids, big carbohydrates into smaller disaccharides and monosaccharides. If you're going down, you're breaking down, what do we call that? Excellent. That is catabolism. So catabolism, if you're breaking down, that is known as catabolism. But let's say you're building up. So when you're building up from micromolecules to macromolecules, if you're building up from nucleotides into nucleic acids, what do we call that now? Excellent. That is known as anabolism. That is known as anabolism. When you are building up is anabolism. When you are building up amino acids to make proteins, that is anabolism. If you are building up glycerol free fatty acids to make lipids, that is known as anabolism. If you are building up monosaccharides, disaccharides, and carbohydrates, that is known as anabolism. So now we established that we have 4 different classes of macromolecules and their respective building blocks, these micromolecules. So we have one is nucleic acids, 2 proteins, 3 lipids, 4 carbohydrates. Do we recognize the 4 different macromolecules? So why do we have catabolism and anabolism? Why do we have catabolism and anabolism? You see, we already established that if you have a macromolecule, and you break it down into micromolecule, what happens? If you break down macromolecule into micromolecule, you're breaking down the bonds. So what's the name of the process where we break down macromolecules into micromolecules? What's the name of that process? It's called hydrolysis. Hydrolysis. Lysis, you're breaking down. Lysis. Hydro is water. So you're using water in order to break down these macromolecules. So why do we break down macromolecules? Let's say we're going from protein to amino acid, from carbohydrates into monosaccharides, from nucleic acids to nucleotides. Why are we breaking down? Obviously, this is catabolism. So catabolism is completely reliant on hydrolysis. But why are we going through all of this? Why are we doing catabolism? Yes, please. So when we break down through hydrolysis, the energy is going to come out. So ATP is going to come out because we need it. When you're catabolizing, you're breaking things down. When you break the bond, remember in chemistry, when you break the bond, you put heat in. When the bond is broken, energy comes out. So when the energy came out, then we're going to power our enzymes. We're going to power our enzymes for our survival and reproduction. So every catabolism reaction is always gonna liberate and exit energy. What do we call that? Exergonic. So catabolism is exergonic. Ergonic means related to energy. Ergonic energy. Exergonic means the energy exited. The energy exited from where? From the bonds of this molecule we're catabolizing into our body. So we catabolize, we break down, and ATP is going to come out, and that is known as exergonic. What about the opposite? So for example, let's say you had a breakfast, bagel, you had pancake. So now, are you going to be breaking down the food, or are you going to be building it up? Please think about it before you answer. If you had a breakfast just now, 5 minutes ago, and you're just sitting down and lecture, you're not running on a treadmill, you're not breaking it down, what do you think is happening? Is the food you ate being in your bodily tissues, is it going to be built up, stored, or is it going to be broken down and burned? Build up. So there's going to be anabolism. So now we're going to go from macromolecule to micromolecule, or from micromolecule to macromolecule. If we want to build up, we go from micromolecule to macromolecule. This is known as anabolism. So let's compare now and contrast. Catabolism and anabolism are not the same thing. Catabolism is when we break down macromolecules, and anabolism is when we build up the macromolecules. Are we clear? Catabolism relies on hydrolysis. Catabolism relies on hydrolysis. If catabolism relies on hydrolysis, what does anabolism rely on? What does anabolism rely on? If catabolism relies on hydrolysis, what's the opposite of hydrolysis? Think about it. Hydrolysis, lysis, breakage. Hydro, using water. So we use water to cleave and break the bonds. Add one OH from the H2O to one side and H to the other side. What's the opposite of hydrolysis? Dehydration is when you remove the water to link them up. Dehydration. Dehydration is when you break down, is when you add the water in order to break down the different parts. And if catabolism is gonna make energy exit out, what do you think anabolism gonna do? Yes? Yeah, it makes the energy go in because we're gonna store energy. In catabolism, energy goes out. So that's exergonic. But in anabolism, energy goes in. What do we call that? That's an endergonic. Excellent. That's an endergonic reaction. So that means ATP is not coming out, ATP is going in. So you need to know that. You need to know that catabolism and anabolism are not the same. Catabolism relies on hydrolysis, while anabolism relies on dehydration. And in catabolism, ATP comes out, and in anabolism, ATP goes in. And we go in different directions. So today we're going to learn about that. We're going to learn about, your cells, your tissues. When you eat pancake, when you eat steak, your tissue doesn't get steak, doesn't get pancakes. It is all broken down in the stomach with the acid and the churning and the mechanical digestion and the chemical digestion and in the small intestine. And it just becomes very mushy, and the food is becoming very soft chyme. And then we broke it down from macromolecules into micromolecules. Then inside the cells, those micromolecules inside the cell, those nucleotides, micromolecule, amino acid, micromolecule, glycerol, micromolecule, free fatty acids, micromolecules, and monosaccharides, micromolecules. Those micromolecules are going to be shuttled in a metabolic pathway where we're going to break them down and get energy from them. If we need energy, but if we don't need energy, they're going to be shuttled into a different metabolic pathway where the energy is going to be stored, not broken down. So let's look into the slides and find out more about this. I did not press record.
Recording in progress.
Okay, so your body weight is determined by the body's energy balance. So if you want to lose weight, you have to burn down the calories, because if you are in the direction of catabolism, it means that there is energy expenditure. So there are two sides of the spectrum. So we have two sides of the spectrum. So there is a nutrition balance. We have to be in a state of nutritional balance. So we have two opposite extremes. The first extreme is obesity, where there is severe high calorie intake. There is severe obesity, and that is represented by high calorie intake. That's one side of the spectrum. On the opposite side, you have minus, and that is malnutrition, and that is there is a deficiency in calories. There is a drop, low calorie intake. Are you supposed to be one of those sides in plus or minus? You're supposed to be right in the middle, where there's an equal calorie intake to calorie burning. But if there's an imbalance and you are now tipped like this, there is more burning for calories than intake of calories, you're going to lose weight. You're going to have malnutrition. And if it's tipped the other way, where there is a lot of calorie intake and a loss and not enough burning of these calories, you're going to suffer from obesity. And both are bad. You cannot favor nutrition or malnutrition over obesity. You can't favor obesity over the malnutrition. So there's a set point in your brain at which your body weight is going to be maintained as you get older. So you see that you feel hungry until you fulfill that particular, you're hungry until you fulfill that particular set point for your weight, and then you oscillate around it. You lose a pound, you gain a pound, but you're not going to drastically lose all of this. And this is regulated, so the control of the weight involves several peptide hormones and regulatory pathways that control the short-term and the long-term appetite. What's the difference between appetite and hunger? Hunger is the drive and the urge that you need to intake calories. The drive and the urge that you need to intake the calories. But appetite is your dietary choice. Do you go to the salad bar or do you go to the other bar? That's your appetite. So how do you choose to fulfill that particular nutritional deficiency? And this is all regulated by hormones that make you hungry. So the people who do the gastric sleeve, they cut a large part of their stomach. So if you look at the stomach, a large segment is going to be chopped off, and when you remove this part, they're not going to feel as many contractions. And the stomach also has cells here, cells inside the stomach that are responsible for secreting these peptides. That's why they call them gut-brain peptides. Gut-brain peptide. Those gut-brain peptides are the ones that are responsible for your regulating your appetite and regulating your hunger stimulation. Now we're going to talk about ghrelin. Ghrelin is secreted from the parietal cells in the fundus. What else do the parietal cells secrete? We talked about this last time we met. Parietal cells secrete what? HCl, excellent, hydrochloric acid. And this ghrelin produces a sensation of hunger and stimulates the hypothalamus to secrete growth hormone releasing hormone. And here you have ghrelin secretion ceases within an hour of eating. Peptide gamma is secreted by the neuroendocrine cells of the ileum and the colon, and they can sense that the food has arrived in the stomach. The primary effect is to signal satiety and terminate the eating. The polypeptide gamma is different from cholecystokinin, CCK. Cholecystokinin is secreted by the neuroendocrine cells in the duodenum and the jejunum. Stimulates secretion of the bile and the pancreatic enzymes, and they stimulate the brain and the secretion of the vagus nerve. So if you look at amylin, for example, amylin is an example of a short-term regulator of the appetite, and it's secreted from the beta cells of the pancreas. Remember we discussed pancreatic amylase, pancreatic lipase, and it produces the feeling of the satiety. So some hormones make you feel like you need to intake calories, you're so hungry, and some hormones make you feel like you need to stop intaking any calories, and you feel satiety. That's why there is pills that people take now if they suffer from bulimia and they want to eat a lot. They take those pills. Those pills stimulate the secretion of the hormones that make you feel full, even if you're not full. There we go. I like this figure a lot because it shows you the relationship between these hormones. Everybody please look at this figure. This figure shows you the gut-brain. Gut, your gastrointestinal tract, brain relationship. You see that the hormones that are in green color, those are the ones that plus, meaning they make you hungry. If plus ghrelin or plus leptin are secreted, you're going to feel hungry. If you inject a rat with ghrelin or you inject a rat with leptin, that rat is going to be eating like crazy. And red is polypeptide gamma, cholecystokinin, and insulin. Makes perfect sense why insulin makes you feel full. Because when is insulin released? Insulin is released when there is high blood sugar. So you already have high blood sugar. You don't need any more. So that's why insulin makes you feel full. Now, this figure not only shows you who makes you feel hungry and who makes you feel full, it even shows you where are they secreted from and where do they target. For example, ghrelin is secreted from the parietal cells of the stomach. Where does it go? It goes to your hypothalamus, and you have to know that the hypothalamus has two offices. Please remember, this is very, very important. There's two offices in your hypothalamus in the brain. The first office is called melanocortin secreting office. This melanocortin secretion, whenever it is stimulated, you're going to feel full. No matter how delicious the food looks, ice cream, pancakes, no matter what they are, I've had enough. Thank you. That's when the melanocortin secretion center has been augmented and activated and initiated. Who activates and augments melanocortin secretion neurons? This is your leptin. So you're going to feel full. That's why many people that have obesity is not because they eat a lot. It's because they have a genetic mutation. They have a congenital disease where leptin is not made. This leptin hormone, they can't make it. So because they can't make leptin, that means they will never feel full. Because leptin is the one who initiates satiety and feeling full. Bless you, Uncle. The other office is called neuron peptide gamma secreting neurons. Those ones, when they are stimulated, you're going to feel hungry and you want to eat the whole table. So who initiates the hunger? You have ghrelin initiates hunger because it stimulates the neuropeptide gamma secreting neurons to make you feel craving food and make you feel hungry. But polypeptide gamma and cholecystokinin, polypeptide gamma is secreted from the large intestine, and cholecystokinin is secreted from the small intestine, and both of those inhibit your neuron peptide gamma secreting neurons, and they inhibit your sensation of hunger. They make you feel full. So leptin, so if you're asked on the exam, how does leptin operate when compared to polypeptide gamma, cholecystokinin, and insulin? You would say that they achieve the same thing with different mechanisms. Well, leptin, polypeptide gamma, cholecystokinin, and insulin, the four of them make you feel full. But they make you feel full in a different way. Leptin goes to the melanocortin secreting neurons to make you feel full, while polypeptide gamma secreted from the large intestine and cholecystokinin secreted from the small intestine and insulin secreted from the pancreas, they inhibit the hunger center. They don't activate the satiety, but they indirectly inhibit the hunger centers, stimulating concomitantly this feeling of satiety. While ghrelin is going to activate the neuron peptide gamma for to make you feel the hunger. This is a very important figure, you need to sit down and read it and examine it in order for it to make a little more sense. So hunger is also stimulated partly by the gastric peristalsis. So we discussed in the digestive system lecture that when food is inside the stomach and is pressing and distending your stomach, there's going to be stretch receptors, nerves around your stomach that are going to be stimulated to make you feel that you're hungry and you need to eat. But appetite can be satisfied by chewing, and lasting satiety depends on nutrients entering into the blood. So the problem is, if you want to be on a diet, you want to eat healthy, you have to be smart and know how your body works so you can use it to your advantage. So whenever you feel hungry and you just want to go and eat a sandwich, that's not good, because you can start chewing on something and that will ultimately make you feel full. Also, drinking. If you drink water when you're hungry, that lowers the calorie intake. And there are plants that once you start chewing on them, they will ultimately make you feel full. And there's some substance that is secreted from the plant that's going to stimulate the satiety response. And sometimes you can eat a really small cheeseburger that has more calories than somebody who eats 5 bowls of salad. Well, that is not economical. That is not smart, because that person is full, eating healthy food, while you are eating junk food, and you will still be hungry again 5 minutes later. So obesity is having body weight that is more than 20% of the recommended weight. So you need to know your body mass index. Many people don't know their BMI, their body mass index. Your healthy body mass index is from 20 up to 25. That's healthy. If you're 26 up to 30, that makes you obesity type 1. If you're from 31 to 34, obesity type 2. If you're from 35 to 40, obesity type 3. And then 40 and above, that is morbid obesity. That is where the patients need a surgery, like the bypass surgery and the gastric sleeve. Bypass surgery is different. Bypass surgery is when they reroute your whole digestive tract. So they're going to go into your stomach and they're going to make your stomach like an egg size, really tiny stomach, and then connect part of your intestine to the stomach and reroute the food. So now your stomach is so small. So when you eat something, you only eat just a little bit and you're gonna feel full. And once you felt full, you're never gonna be hungry again. So you're never gonna have obesity at all. So that's like a solution that, you never come back from that. For gastric sleeve, people return and gain weight again. But people who did the bypass, they never gain weight ever again. Is that clear? Questions? So this is a MRI image of a person with obesity and a person with normal weight. You see all this yellowish, buttery adipose tissue? Now, calories are a measure of the capacity to do the biological work. So 1 calorie is the amount of heat that is required to raise the temperature of 1 gram of water by 1 degree Celsius. That's the scientific definition of a calorie. Calorie is how much, heat is a form of energy. We have electrical energy, we have kinetic energy, we have thermal energy. So the thermal energy that is hot enough to make 1 gram of water gain 1 degree Celsius hotter than before, that's a calorie. And 1,000 calories, known as a kilocal, is represented by a capital C, because people are unaware now. They find some food and they see that it's 150 calories. No, these are not 150 calories. These are 150 capital C. So that means this is 150,000 calories. It is not 150 small c. Now that's 150 calories. But if it's 150 capital C, that is 150 actually thousand calories. Nutrients are any ingested chemicals that are used for growth, repair, and maintenance of the body. There are 6 classes of nutrients. So we have macronutrients and micronutrients. What's the difference between macronutrient and a micronutrient? Macronutrient, you need to take it in large amounts. That's why they call it macronutrient. Micronutrients, you don't need to take that much of it. You just only need to take a little bit. So the 6 classes of nutrients, we discussed those. Those are the carbohydrates, the lipids, and the proteins. And these are considered macromolecules because they must be consumed in relatively large quantities. But vitamins and minerals, these are micronutrients that are to be taken in only small quantities. So the functions of the sugars. There are structural components of other molecules, including nucleic acids, glycoproteins, glycolipids, ATP, and related nucleotides. So if you look at the sugar, sugar is an important component of, remember, glycoproteins at the surface of the cell membrane that is detected by your immune system for immunosurveillance. Glycoproteins, glycolipids, you remember that? Glycoprotein, glycolipids, ATP. So we need to regulate the sugar index in our body. We don't want hyperglycemia and we do not want hypoglycemia. So hyperglycemia is when there is so much sugar in the blood level. There's so much sugar in the blood. So hyperglycemia is an increase in blood sugar. Too much sugar. And hypoglycemia is when there is too little sugar. That is hypoglycemia. In hyperglycemia, the chemoreceptors are gonna send information to the pancreas. And the pancreas is gonna secrete, if there's too much sugar, it's gonna secrete insulin. And insulin is going to lower the blood sugar, returning back to homeostasis. If you have a drop in blood sugar, the information is going to be sent to your pancreas again. And the pancreas is going to secrete glucagon. And that is now going to increase the blood sugar. Because insulin is an anabolic hormone, it built up the sugar. while glucagon is a catabolic hormone that breaks down the sugar. Lipids. These are structural macromolecules that are non-polar. So carbohydrates are polar. They dissolve in water, while lipids are non-polar. They don't dissolve in water, they dissolve in lipids. That's why they are an integral part of your plasma membrane. So once you eat a meal that has a mixture of like a cheeseburger that has both lipids and carbohydrates, we need to separate those two and send the carbohydrates to be digested in the glycolytic pathway to get ATP, and send the lipids separately in a different catabolic pathway known as beta oxidation pathway. So if you look at the daily requirements of fat should be less than 35% of the daily calorie intake, because less than 10% of the fat intake should be even saturated. Saturated fats are very bad for you. So most fatty acids are synthesized by the body, but the essential fatty acids must be consumed. Saturated fatty acids come from the animals, like the animal meat, the egg yolk, the dairy products. But unsaturated fats, these are taken from the vegetable oils, the seeds, and they are found in the nuts. Cholesterol is found in the egg yolk, the cream, the shellfish, organ meats, and other meats, only in tiny trace amounts in foods of plant origin. Lipoproteins. These are complexes that transport lipids in the plasma, and there are tiny droplets with the core of cholesterol and triglycerides. So if you look at the name, lipoproteins. What is a lipoprotein? What is a lipoprotein? So from the name, if we look at a lipoprotein, lipoprotein is a two-in-one. Lipid, lipo for lipid. Protein is obviously protein. So that's like a submarine. A submarine, because from the outside is a protein. P, protein, P, protein, protein, P for protein, 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 protein. On the outside is protein, but on the inside it is not a protein. On the inside it is butter, yellow lipid. On the inside it is butter, yellow lipid. L for lipid, L for lipid, L for lipid, L for lipid. You see? Are we clear? So this is how, because you know the blood is polar. The blood is polar. And the proteins are polar. So the outside of the lipoprotein is going to be dissolved in the polar blood, while the core lipid is going to be dissolved in the middle and the core of the lipoprotein. And that is just like how we enter a submarine and go down in the water. The outside of the submarine can swim in the water, while us inside the submarine, we're going to be surviving in the core. So now let us look at, there we go. That's an example of a lipoprotein. That's what we just drew here. On the yellow, on the outside, what do you see on the outside? Those are the phospholipids. Are phospholipids polar or non-polar? Phospholipids. Are they polar or non-polar? Phospholipids are, you know, they have a phosphate head. And the phosphate head is polar or non-polar? It's polar. So from the outside, the submarine is dissolved in the blood, in the water, but deep in the core, what do you have green in here? This is the lipid. So now they look at lipoprotein, they divided the proteins into different structures. Divide the proteins into different structures. So there is a chylomicron. What's a chylomicron? A chylomicron has a little bit of phospholipids, a little bit of proteins, but it has a lot of triglycerides. It has a lot of triglycerides. If you look at the very low density lipoproteins, it's just like a chylomicron. The only difference is the very low density lipoprotein has a little more cholesterol than does the chylomicron. But if you look at the low density lipoprotein, that's the one that makes the atherosclerosis. That's the one that makes the heart attack. What does the low density lipoprotein have? It has a lot of cholesterol. A lot of cholesterol. And if you look at the protein, you see that there is a high protein content in the HDL, then cholesterol, and then the triglycerides. Cholesterol and the serum lipoproteins. So there are three pathways by which the lipoproteins are made and processed. There is the chylomicron pathway, and we discussed that the chylomicron pathway, you would have the chylomicrons forming in the absorptive cells of the small intestine, and they enter the lymphoid system, then they enter the bloodstream, and there is a lipoprotein lipase, which is in the endothelial cells that hydrolyzes triglycerides into monoglycerides and free fatty acids. Resulting free fatty acids and glycerol enter the adipocytes to be made into triglycerides for storage. So the chylomicron remnants, these are the remainder of a chylomicron after the triglycerides have been removed and we exploited them for the metabolism purposes. If you look at the very low density lipoproteins, comparing them to the low density lipoprotein pathway, the very low density lipoproteins are produced by the liver in order to transport the lipids to the adipose tissue. Then when the triglycerides are removed in the adipose tissue, very low density lipoproteins are going to be converted into low density lipoproteins, and they're going to contain most of the cholesterol. Low density lipoproteins are absorbed by receptor mediated endocytosis by these cells that are in need of cholesterol. So if we look at this very interesting figure, we see that the chylomicron pathway is made by, you have small intestine, the lymph, remember the lacteals? The lacteals are these appendages that are found in your small intestine. And these lacteals are important for absorbing what? This is a lymphoid tissue. What are lacteals? Who remembers what are lacteals? Anybody remembers lacteals? What are lacteals? Lacteals are lymphoid aggregates that are found in the small intestine. So if I tell you they're found in the small intestine, you should now start to get a hint of what they do. Because what does the small intestine do? It absorbs the food. So the lacteals are absolutely going to be absorbing. But what are they absorbing? What are the lacteals absorbing? Yes, they are absorbing fats and lipids, not proteins. So there we go. You see the lacteals absorb the lipids. Then the lymph is going to drain the lipid into the bloodstream. Then the lipoprotein lipase is going to remove the lipids from these chylomicrons, and we're going to deposit the lipids and store them in adipocytes used for other cells, and then the liver is going to dispose these chylomicron remnants. Then once the chylomicrons have been sequestered in the liver, then you see that very low density lipoproteins are going to be produced by the liver. And in the very low density lipoproteins, if you look at the structure, you see that very low density lipoproteins have a lot of triglycerides and some cholesterol. So you see here, triglycerides are going to be removed and stay in the adipocytes. And then when you remove the triglycerides, when you remove these triglycerides, you're going to wind up with a lot more cholesterol that's left. The left cholesterol now is going to convert for us. This what used to be the very low density lipoprotein is now going to be converted into low density lipoprotein. So the cells are going to absorb the low density lipoproteins by the receptor mediated endocytosis, because we want to bring in this globule that is full in its core with a lot of lipids and perform beta oxidation. This beta oxidation is going to harvest for us ATP from this lipid that we have absorbed. So the liver produces empty high density lipoprotein shells. These high density lipoprotein shells are going to pick up the cholesterol and the phospholipids from the tissues. They're going to be filled, these filled high density lipoproteins return to the liver, and then the liver is going to excrete the excess cholesterol and bile salts. So cholesterol and serum lipoproteins, these are desirable to maintain the total plasma cholesterol, and most of the body's cholesterol is endogenous. You know, cholesterol is very important because cholesterol is part of the plasma membrane. It's important for the stiffness and the structural integrity of the plasma membrane. And this cholesterol has to be taken in moderate amounts. So if you have excessive amount of cholesterol intake that far exceeds and surpasses your required daily dosage to supply the synthesis of the plasma membrane, then that is going to build up in the blood vessels of your coronary circulation, which is going to lead to heart attacks. Cholesterol is also important for what else? For hormones, for essential hormones. There are cholesterol-based hormones. These are cholesterol-based hormones. These cholesterol-based hormones give us estrogen, progesterone, testosterone. So it's interesting. All of these hormones, these essential hormones, they are made from cholesterol. All of them. Progesterone, testosterone, estrogen, estradiol, they're all made from cholesterol. Now, when we move to proteins, we see that proteins constitute from 12% to 15% of the total body mass of your body. And 65% of it is found in the skeletal muscle. So your proteins are stored in your skeletal muscle. Remember, you guys last semester did the skeletal system and the muscular system. You went over the neuromuscular junction and the troponin and the tropomyosin and the actin and the myosin filaments. What are those? Those are proteins. So your muscles, skeletal muscles, and the cardiac muscles and even the smooth muscles. They all have proteins, and these proteins are responsible for the muscle contraction once a membrane voltage change is mediated by the passage of these action potentials. That's going to lead to the muscle contraction. Also, if you look at the cilia, remember when we discussed cystic fibrosis? In cystic fibrosis, that's a disease where chloride channels are no longer made on the mucosal layer of your respiratory tract. If there is no chloride channel, we're not going to be able to absorb chloride. So sodium is going to stay in our mucosa, and water is going to move from an area of a low sodium concentration to an area of a high sodium concentration in the lumen, and that's going to cause a mucus plug. A mucus, that's going to cause a mucus plug in the respiratory tract. So the requirements for protein intake required daily assessment is 46 to 56 grams per day. So higher intake is recommended under conditions of stress, infection, injury, and pregnancy. And excessive intake is going to overload the kidney with nitrogenous waste and can cause kidney damage. There are 8 essential amino acids. Essential meaning that you need to take them from an exogenous outside source. They cannot be synthesized by your own body. Like isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Those, you have to take them from the food that is ingested. But the other inessential amino acids, they can be made by your own body. That's why if you have a deficiency of these 8 essential amino acids, you're going to have a problem making essential hormones and essential proteins, essential enzymes. You know these enzymes? What are the enzymes? Enzymes are proteins, and proteins are made of polypeptides of amino acids. Some of these enzymes are made up of polypeptides of essential amino acids. So if you don't eat them and you have malnutrition and these proteins are not completed with these missing essential amino acids because you're not eating the right foods, then you're going to have a deficiency in these proteins, enzymes. And even though ATP is there, the enzyme is not going to be working because some very essential amino acids are missing. Tryptophan is found in chicken, and tryptophan is very important because it makes hormones like dopamine, and you know the rich source for these essential amino acids are in the animal meats. So many people who are vegetarian for different purposes, different reasons, they always have a problem with the replenishing of these essential amino acids. Minerals and vitamins. So minerals and vitamins, these are not used as fuel, but they are supplementary, auxiliary, supportive, and they're essential to the ability to use the nutrient. They most cannot be synthesized by the body, but they are found in the diet. And vitamins, we only need small amounts of them, not too much, because they work as cofactors. These very important micronutrients, these very important minerals and vitamins are being used like cofactors in the enzymes that participate in redox reactions, lose electron oxidation, gain electron reduction. This is all facilitated and made possible by the functions and activities of the minerals and vitamins. So these minerals are chemical elements which are mostly metals, and the plants extract them from the soil. So minerals are taken from the soil, and when you eat the plant, you're going to eat the minerals that the plant took from the soil. And minerals constitute 4% of the body mass. 3/4 are calcium and phosphorus in the bones and the teeth. Phosphorus is a key structural component of phospholipids: ATP, cyclic AMP, GGP, and creatine phosphate. And that's the basis for the phosphate buffer system. So the phosphate is an example of a mineral that's very important. You know, adenine triphosphate. Some enzymes, if you add a phosphate to them, they get turned on, they get activated. Some enzymes, you remove phosphate, they stop working. So that shows you how important phosphate is. Adenine triphosphate. Cyclic AMP, adenine monophosphate. Cyclic AMP is the second messenger that is going to mediate an internal signal transduction pathway that's going to activate a target enzyme that's going to go and give us the target effect of some hormone that couldn't enter into the cell and had to bind an outside extracellular receptor. Other examples of minerals are calcium, iron, magnesium, manganese, that function as cofactors for the enzymes. Vitamins are organic nutrients that participate in metabolic reactions. Some vitamins are synthesized from precursors called provitamins. Now let's begin with the first class of macromolecules, and let's look at how your cell is going to make ATP out of this micromolecule. So now we're not talking about digestion anymore, because we already ate the food, ingested the food, digested the food physically, mechanically, and secreted enzymes and acid, and then we transported, absorbed the food from our intestine into the blood stream. So now let's begin with the first of the four. That's the glucose metabolism. Glucose metabolism is accomplished in small steps. Each step is controlled by a separate enzyme. It's like a dominoes effect. So the first enzyme does the first job and then passes the torch over to the next successive enzyme in this series of transduction cascade steps known as the glycolytic pathway. So in which the energy is released in small, manageable amounts and as much as possible. It's transferred to ATP and the rest is released as heat. Glycolysis. Glycolysis, you would have glucose split into two molecules of pyruvate. Glucose is split into two molecules of pyruvate. This is the major first 1/3 of the glycolytic pathway, and that is known as glycolysis. This is glucose is broken down from 6 carbon molecule into 2 3 carbon molecule. Like a $10 bill, you break it down into 2 $5 bills. Then, how many carbons are in glucose? Glucose is comprised of how many carbon atoms? 6. So we're going to break down the 6 carbon glucose into how many? 2, 3 carbon molecules. What do we call each of the 3 carbon molecules? Pyruvate. So we had something like this. If you look at the glycolytic pathway, one, 2, 3, 1, 2, 3. So that's a 6-carbon glucose. When we go through glycolysis, we're going to end up with 2 3-carbon molecules known as pyruvate. That's a 3-carbon pyruvate, and another 3-carbon that's the first part, known as glycolysis. Then there is 2 routes. After we've broken down glucose into 2 3-carbon molecules known as pyruvates, we're going to convert that into is there oxygen or is there no oxygen? That's the question. To be or not to be? That's the question. The question is, after we've broken down this 6-carbon molecule, glucose, into 2 3-carbon molecule, pyruvates, is there oxygen around? If there is oxygen around, that is going to be known as aerobic metabolism. If there is no oxygen around, minus oxygen, that is going to be known as anaerobic metabolism. So anaerobic fermentation reduces pyruvate to lactate without using oxygen. And aerobic respiration requires oxygen and oxidizes pyruvate to carbon dioxide and water. Now, if you look at the actions of the coenzymes, so the coenzymes are enzymes that remove electrons, such as hydrogen atoms, from the intermediate compounds of these pathways. So the enzymes are going to transfer the hydrogen atoms to the coenzymes, and the coenzymes and these cofactors, they're going to donate them to other compounds later in the reaction pathway. There are 2 coenzymes of special importance, and that is FAD plus and FAD. So if you look at NAD plus, nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. It has a plus charge on it. So you can tell, is that an oxidizing agent or is that a reducing agent? If it has a plus charge on it. If there's a plus charge on it, it means that it lost a minus electron. Remember, the cells are not like us. If you look in your bank account and you find a plus 1, it means that you gained $1. But if you find a minus, it means that you got withdrawn. You lost. So somebody with a minus bank account is poor. Somebody with a plus bank account is rich. That's not the way the atoms operate, because the atoms don't work in dollar. They work in minus electrons. So if you give me 1 minus electron, I am rich. And you lost a minus, so that means you're plus. That means you're poor. If I have minus 2 and you only have minus 1, I am more rich than you as an atom because I've gained 2 electrons. That's why I have 2 minus electrons, and you only have 1 minus electron. You're poorer than me. And my 1 minus electron is a lot better than somebody who has plus, meaning that they lost an electron. Compare that to somebody who has plus 2, plus 3, plus 4. That means the more pluses you have, the more poor and deficient in electrons are you. The more minus you have, the more rich you are. So if you look at nicotinamide adenine dinucleotide plus, that means it has lost an electron. So if it has lost an electron, Leo says GER, lose electrons, oxidation, gain electron reduction. So when you're oxidized, it means that you are a reducing agent. Reducing agent, that means you would help others gain electrons. FAD, flavin adenine dinucleotide. And that one is going to accept 2 hydrogen protons and it's going to be the oxidizing agent. So hydrogen atoms are going to be removed from the metabolic intermediates in pairs. So 2 protons and 2 electrons at a time. They are transferred to a coenzyme, and this produces a reduced coenzyme with a higher free energy content than before the reaction. The coenzymes become temporary carried of the energy extracted from the glucose metabolites. So if you examine the glycolytic pathway, this is what we were saying earlier. Glycolysis and anaerobic fermentation. If you look at the mouse, we have a 1, 2, 3, 4, 5, 6 carbon molecule. What is that? That is glucose. So in order to break down glucose into, you see what do we have in the end? We have 1, 2, 3 carbon pyruvate and 1, 2, 3 carbon pyruvate. So what happened with the glucose and in the first step? We lost ATP in order to add a yellow phosphate group to the glucose. Then this process is called phosphorylation. That's an investment phase. We invested a phosphate atom on the glucose that is to be catabolized into 2 separate 3 carbon pyruvate molecules. Then we added another phosphate group to the 6 carbon glucose molecule, and then we have a 6 carbon glucose molecule that has on both sides phosphates, and then we break that down into 2 pieces, and then we perform oxidation. The oxidation now is going to allow us to harvest and get back our ATP. And once we have the pyruvate, everybody please look at where my mouse is. That's the terminal point. That's the end of glycolysis. We already successfully went down from 6 carbon glucose molecule into 2 3 carbon pyruvate molecule. And the 2 pyruvate carbon molecules are we're now going to look and see, is there oxygen? And if there's oxygen around, then we're going to perform, if there's oxygen around, we're going to make ATP. If there is no oxygen around, then we're going to perform anaerobic fermentation. What's the highlight of anaerobic fermentation? This is very important, guys. If you look at aerobic respiration, the highlight of aerobic respiration is that it utilizes oxygen where? In which organelle do we perform this aerobic respiration? In the mitochondria. And this anaerobic fermentation, the highlight of it is that it gives us lactate. This is why when you're running on a treadmill, you feel the acid in your knees and you feel the acid in your muscles. Why? Because the lactate is being secreted. Priming. Glucose 6-phosphate is rearranged and isomerized to form fructose 6-phosphate. And the phosphor is phosphorylated again to form fructose 1,6-diphosphate. And then we perform cleavage, which is small changes are going to result in molecules of phosphoglyceraldehyde. The result of cleavage is 4 ATPs produced. But remember, there was an investment phase. We invested 2 ATPs in order to start up glycolysis. Then in the end, when we get 4, we return the 2 ATPs we invested, and then we made up 2 ATPs as a result. The fate of lactate. Lactate leaves the cells that generate it and travel to the liver via the blood. When oxygen becomes available, The liver oxidizes it back to pyruvate, and oxygen is required for this. This is the part of the reason that we breathe more vigorously after the exercise. We discussed that. Now, in aerobic respiration, we are now at the stage where we have our 3-carbon pyruvate ready after cleaving this 6-carbon glucose molecule. So in the presence of oxygen, pyruvate is going to enter the mitochondria and is oxidized by aerobic respiration. Most of the ATP is generated through aerobic respiration, and it requires oxygen because oxygen is the final electron acceptor. So the aerobic respiration is going to occur in 2 principal steps. There's the matrix reaction, and their controlling enzymes are in the fluid of the mitochondrial matrix, and membrane reactions, their controlling enzymes are bound to the membranes of the mitochondrial cristae. The mitochondrial matrix reactions, you see that this is the pyruvate, and then we shuttle pyruvate, we convert pyruvate into an acetyl group. You see, acetyl has 2 groups just like pyruvate. Then we shuttle pyruvate into this mitochondrial matrix reactions to ultimately make our ATP. The matrix reactions, some of the energy in the 4-carbon substrate is going to go to phosphorylate guanosine diphosphate. I convert it to guanosine triphosphate. The 2 final electron atoms removed, these are going to be transferred to NAD+. So mitochondrial electron transport chain represent a series of compounds that carry out the series of processes in which we are utilizing a protein motive force and hydrogen protons on influxing between the outer and the inner membranes of your mitochondria, and that leads to the spinning of the rotor, this rotor known as the ATP synthase. Now, the membrane reactions, known as we have the flavin mononucleotides, and this is an acceptor of electrons from NADH. So if we look at the figure, this is an excellent figure because it shows us how does the mitochondrial electron transport chain operate. So we begin with this FMN reducing agent that is going to remove an electron in the form of a proton from NADH, and that's the first enzyme complex. And we're going to be passing electrons through the coenzymes and the cytochrome C enzymes until we finally arrive at the oxygen, which is the final electron acceptor. In this process of shuttling and passing electrons from one electron donor to the next electron acceptor, the energy is going to be passed. And the passage of the energy is going to lead to this spinning of the rotor that we will see now. So let us now look at this chemoosmotic mechanism where we have a proton motive force. Proton motive force. This proton motive force is responsible for funneling and channeling hydrogen protons into the ATP synthase. So the chemoosmotic mechanism, you would have hydrogen proton that is currently rushing back through the ATP synthase, and channels derive the ATP synthesis. So there we go. This is a very important figure. Everybody please pay attention to this very interesting figure. So what is happening in this figure that we're looking at. This is the chemiosmotic mechanism of ATP synthesis. So in this figure, we are making ATP. How do we make the ATP? We make the ATP utilizing, so where is this happening? Where is this chemiosmotic mechanism, ATP synthesis happening? In the mitochondria. Now, excellent. Where exactly in the mitochondria? Before you shout your answer, look at this box that is showing you a cross section. How many phospholipid bilayers do you see? 2. There's 2 leaflets here and 2 leaflets here. So those are 2 membranes, 2 plasma membranes. Each one has a double layer, an inner leaflet and an outer leaflet. The one on the inside, that's the inner membrane, and that is the cristae, the inside of the mitochondria. And this is the outer membrane of the mitochondria. Now let's look at what happens. So what happens is there is a constant active pumping of hydrogen protons. From which direction? From the inside to the outside. You see? So those 3 enzyme complexes, 1, 2, and 3 only, not the last one, not the ATP synthase. 1, 2, and 3, enzyme complex 1. Does everybody say enzyme complex 1? Everybody say enzyme complex 2? Does everybody say enzyme complex 3? Those 3 enzyme complexes have something in common. They are pumpers. They pump. What do they pump? They pump hydrogen protons. They pump hydrogen. You see? They are pumping H+, H+, H+. They are pumping hydrogen protons to the outside. Why are they pumping hydrogen protons from the inside of the inner membrane to the outside? Because they're trying to create a gradient, a proton gradient, a proton motive force. Here, this proton motive force is being created by these 3 enzymes. They are removing hydrogen proton from the inside of the plasma membrane to the outside. Now what are you going to have here? You're going to have a build-up of a lot of hydrogen protons in the intermembrane space between the inner membrane and the outer membrane. Now what's going to happen? Now we come to our 4th and final important protein. You see this here, ATP synthase? All of the hydrogen proton that has been accumulated here because of these enzyme complexes pumping out hydrogen proton is gonna now go back in the middle through this ATP synthase, spinning the ATP synthase, rotating like a rotor. So this ATP synthase spinning, and when the ATP synthase spins because of hydrogen protons pumping inside it, what do we get? We get ATP. Now you see how we make ATP? We pump out hydrogen protons with these enzyme complexes, and then the hydrogen protons that we pumped out, they're gonna flow in through the ATP synthase rotor, spinning it, and then when it spins, it makes the ATP. So if you look at the summary of the sources of ATP that are generated by the complete oxidation of glucose. So we oxidize glucose through glycolysis, and we get the 2-pyruvate. Remember we discussed that. We get our 2-pyruvates. And this process gives us a net 2 ATP. It doesn't give us 2, it gives us 4 ATP. But remember, we invested 2 ATP molecules. Then the 2 net are going to be secreted, and then there's a citric acid cycle. Citric acid cycle is going to spin the rotor, and then 2 ATPs are going to be generated, 3 ATPs, and then 25 ATPs. These are the final byproducts. Glycogen is an energy storage molecule. So ATP is quickly used after it's formed. And you need to know the difference between glycogenesis and glycogenolysis. What's the difference between glycogenesis and glycogenolysis? What's the difference between glycogenesis and glycogenolysis? Glycogenesis and glycogenolysis. Glycogenesis, from the name, genesis, the making. So glycogenesis means we make glycogen. We make glycogen. We make glycogen. So is that catabolic or anabolic? That's anabolic. Excellent. We make, that is anabolic. Is this endergonic or exergonic? Endergonic. Excellent, because we're storing energy. That is endergonic. Is this hydrolysis or dehydration? That is dehydration. Excellent. Perfect. Dehydration synthesis. While glycogenolysis, now is not genesis, now is lysis. We are breaking down. So now we have glycogen and we are lysing it, breaking it down into what? Into glucose. Here we had glucose, we break it down and we build it up into glycogen. But here we have glycogen, we break it down into glucose. So is this process anabolic or catabolic? That's catabolic. Is this process endergonic or exergonic? That is exergonic. Is this dehydration or synthesis? This is synthesis. Also known as hydrolysis. Are we clear? Questions? Questions? So there we go. The major pathways of glucose storage and use is that glucose is in the blood, and in order to bring the glucose from the blood into the liver, what do you notice here? Can everybody tell me what happened to, everybody please look at the mouse. You see what my mouse is? In the mouse I have blood glucose. Once the blood glucose entered this area in blue, what is that? What is the area in blue? This whole blue box, what is that? That's the liver. Red is the blood, blue is the liver. If the arrow points down, that means we went from the blood into the liver. If the arrow points out, it means we went from the liver out to the blood. So what do you see here? When the blood glucose went inside the liver, what happened to it?
It gets a phosphate.
It got an extra phosphate group. What is the significance of that? Why did we add a phosphate group to the glucose we brought in? Why did we add a phosphate group to the glucose we brought in? Because when you add a phosphate to the glucose you brought inside the liver, can this glucose escape back out, go to the blood back again? No, that's called glucose trapping. So we trap glucose by adding a phosphate to it. So now let's say you go and you run on a treadmill. Now you need the glucose back from the liver. What is the liver going to do to liberate and allow this glucose to go back to the blood? The arrow to point out. What's going to happen? Remove the phosphate. What are the enzymes? What is the name of the enzyme that remove the phosphate called? Yes, this one, excellent, is hexokinase because it's doing that in the liver. That's hexokinase. But in general, any enzyme that remove a phosphate is called a phosphatase. Phosphatase. And the enzyme that add a phosphate, the opposite of a phosphatase. What's the name of the enzyme that add a phosphate? What is that one called? It is called a kinase. So you need to understand that when the liver want to uptake glucose and trap it in the inside, does it use kinase or phosphatase? Kinase. And if the liver want to send out the glucose because you need it now for catabolism, is it going to utilize a phosphatase or a kinase? Phosphatase. Now we finished with the sugars. Let's look at the lipids. Lipids are part of our diet too. We don't only metabolize carbohydrates. We don't only metabolize proteins. We also metabolize lipids. So the lipids are broken down by a process known as beta oxidation. This beta oxidation also happens just like glycolysis that happens in the mitochondria. Just like aerobic glycolysis happens in the cytoplasm, in the cytosol. But the aerobic respiration happens in the electron transport chain. The aerobic respiration happens in the mitochondria. And the beta oxidation to break down the lipids is made in the mitochondrial matrix. If you look at proteins, proteins have a two terminal. You need to know what proteins look like. Proteins Proteins have a central alpha carbon. It has a carboxylic terminal. It has a amino terminal. And it has a hydrogen terminal. And it has an or substituent terminal that makes it different. That's an amino acid. If I want to catabolize amino acids, I must perform the interconversion, which is the deamination, the removal of this amino group, and sometimes we perform amination to interconvert it into a digestible precursor for the protein catabolism. Transamination represents the transfer of an amino group from one molecule to another. Now let's look at the pathways of amino acid metabolism in relation to glycolysis and the citric acid cycle. We see that protein and amino acids, we perform deamination. When we deaminate the amino acids, we convert them into keto acids. Remember that. When you deaminate, you devoid an amino group from an amino acid, you turn it into a keto acid. And this deaminated, this amine group is going to go inside the liver, and it's going to be converted into glutamic acid. Glutamic acid. If you now look at the fate of this amino group, we're going to add alpha ketoglutarate acid to this amino group. We're going to get glutamic acid, which is an important amino acid. This glutamic acid can later on, we can remove this deamination. We can remove the deamination, and we shuttle this amine group into the urea cycle in order to make urea. So urea cycle is going to take all the waste. Urea cycle is so important because look at what it does. It takes an amine poison and couple it with carbon dioxide. Is carbon dioxide toxic and poisonous and stressful or not? It's not. Carbon dioxide is not poisonous, stressful, and toxic. It is very toxic and stressful. So it will take carbon dioxide and it will add it to the amine toxic and combine them and make an even worse toxic secreted in the urine. What is the name of that toxic called? Urea. Very good. Excellent. And then he's going to remove it in the urine. The relationship between the glutamic acid and alpha ketoglutarate acid is important because this alpha ketoglutarate acid is going to go into the citric acid cycle, and the citric acid cycle is going to take acetyl-CoA in order to, so acetyl-CoA is a acetyl-CoA is going to enter the citric acid cycle and give us alpha ketoglutarate acid. This alpha ketoglutarate acid is a center of many metabolic processes. You see, we can add an amino group to it, convert it into glutamic acid. We can take glutamic acid and deaminate it and make alpha ketoglutarate acid. So there is a turnover happening. If you look at the keto acids, which were made by removing an amino group from the amino acids to get a keto acid, then these keto acids are, number one, they're going to be used in the glycolytic pathway to make pyruvate out of glucoses. These keto acids are going to be interconverted into acetyl-CoA in order to enter the citric acid cycle and make alpha ketoglutarate acids. The liver is important. That's why alcoholic liver disease is very dangerous. We talked about the importance of the liver in the past. Who can remember the importance of the, when we did the immune system, the liver was important. When we did the heart and the blood, the liver was important. When we did the digestive system last meeting, the liver was important. Remember when we went over the liver? What did we say the liver was important for the digestive system? Why? Remember we said that the liver is right in the middle between the digestive system and the heart. We said that the blood doesn't immediately go back to the heart because the digestive tract is full of microorganisms and a lot of waste. So the blood goes to the portal system into the liver, and the liver is like a sponge filter. It's going to filter the blood, so the blood that goes back to the heart is cleaner. So that shows you the help of the liver to the digestive system. Your immune system is also getting a lot of help from the liver. How? Where are the immunoglobulins that your immune system using to make the antibodies? Where is it made? Is made in the liver. For your heart and your blood. Where do we make the albumin? The albumin, the protein that is transporting a lot of the insoluble, the water insoluble proteins, it's made in the liver. So now we're going to see that the liver plays an important role in addition to your metabolism system. So your carbohydrate is metabolized in the liver, lipid metabolized in the liver, protein metabolized in the liver. So diseases like hepatitis and cirrhosis, from the name, itis, sinusitis, otitis media. When you have itis, that's an example of inflammation. Hepatitis is the inflammation of the liver. That's a very, very, very dangerous disease. So it is caused by 6 strains of hepatitis viruses, HVA to HVF, and the most common form is hepatitis A. But hepatitis B and C, these are more serious, and they can even cause cirrhosis of the liver and cancer. What is cirrhosis of the liver? It's like when the liver becomes so scarred. It's like, you know the candle, ever seen a candle? When the candle melts, you know the thing that is falling to the side of the candle?
The wax?
The wax. Yeah, wax. Your liver is going to become so scarred and waxy. That's cirrhosis. Cirrhosis when the liver becomes waxy. It's like a candle melted on it. Is when the liver becomes waxy. Is that good or bad? That's very bad. So cirrhosis is irreversible inflammatory liver disease with poor prognosis. Nobody goes back from liver cirrhosis. I had a friend of mine, excellent guy. He was just a raging alcoholic. And now he's so young, but his liver is suffering from the wax, cirrhosis. So his liver is not working. That means no immunoglobulins. That means no plasma, blood, no albumin, no plasma albumin. Means the blood that is going out the digestive system is no longer going to be filtered when it goes back into the heart. You see the wax? You see the waxy liver? What do we call waxy liver? Cirrhosis. Can this liver detoxify or cause any albumin synthesis? Can it help your immune system by making immunoglobulins? Can this waxy liver now make the supportive functions and bile and all of that? You know, people think that bile is made in the gallbladder. It's not. Bile is stored in the gallbladder. It's made in the liver, but it's stored in the gallbladder. So all of that, goodbye to that. So who makes cirrhosis? Many things. The hepatitis, the inflammation of the liver, as well as the alcoholism. Drinking the alcohol. That is going to cause severe. That's going to cause severe cirrhosis. And you think, oh, I'm so young, it's never going to happen to me. It happens to the older people. I told you, my friend is young. He's 40. And now his liver is completely done. He cannot digest anything. And is suffering from sadness and depression.
So is he gonna die?
Yeah, unless he buys a new liver. You gotta wait in line. And it's very political. They don't give it to anybody. You know, in the hospital they say, we're gonna wait list for a kidney. Wait list for a liver. Did they select the mayor?
Yeah.
Is he that guy?
Yeah.
He won?
Yeah.
I don't know. I'm not excited about him. I don't know why. Everybody's excited. You know, ever since I voted for Obama, and there was such a big excitement at the time, and he won, and he didn't do anything for us. He's just. I'm no longer. Remember, when I was your age, the big guy like him was Obama. Like when I was your age in college. Oh, Obama, vote for the first black president. He's gonna help everybody. Hope is on the way. I was studying for my exam, waiting in line to vote for him. And then when I got him in there, I'm the one who got him in there. He didn't do anything. Did he do anything? He didn't do anything. So from that day, I don't vote no more. But he won, huh? He won? Yeah. You will see. They will not do anything. I'm sorry to break out the news. They're not gonna help you. Okay. Yeah, just get your education. That's gonna help you. I'm not gonna interrupt my lectures to go vote for someone who will not help me. My lectures help me survive. He's not gonna help me. So I'm just gonna keep my just stay truthful to your job and your work and your career. and you will survive. Right, Divyansh? Okay, post-absorptive state. The post-absorptive state regulates the plasma. The post-absorptive, this is a lot. We have 20 more slides. We can do it. The post-absorptive state regulates the plasma glucose. So post-absorptive is we already ate, we now absorb the food into the into the blood. Now your metabolic rate is going to increase if you're a type of person who is moving and you're exercising. You know we are supposed to be, our body was made for, like, you know, the caveman. The caveman was running for the food every day, but now everything is so technological. So you get to stay in your office from 9 to 5, not doing any work. Another childhood obesity. People, all the cardio they get is 20 steps to the car, drive the car, then 20 steps from the parking lot to the office, then 20 steps back to the parking lot, then 20 steps back to the house, and then laying on the couch. That's why obesity is so prevalent now. Prevalent only in the first world countries, but the poor countries, because their job is not office job, so they need to be working a lot, like construction, so they burn all the food, calorie intake. So the total metabolic rate is the sum of the basal metabolic rate and the energy required for voluntary activities. So we discussed how the body regulates hyper and hypothermia. And enzymes that control our metabolism depend on an optimal, stable working temperature, and that is to maintain this heat loss from happening. Thermoregulation is a balance, homeostasis. We discussed that when your hypothermia is too cold, we shiver in order to restore the proper temperature. Hyperthermia is when we heat up and then we release the sweat in order to cool off. Core temperature is the temperature of the organs in the cranial thoracic cavity. And we discussed that the heat production and loss is regulated by your skeletal system. So the heat is lost through radiation, which is the emission of the infrared rays by the moving molecules. So you see, this is the heat loss by conduction and convection. Convection meaning heat is being moved through the molecules, and convection that is mediated through the air, through a medium. Evaporation is the change from a liquid to a gaseous state, and there's a cohesion of water molecules that hampers their vibratory movement in response to heat input. If the temperature of the water is raised sufficiently, its molecular motion becomes great enough for molecules to break free and evaporate. The hypothalamic thermostat. So there's a preoptic area of the hypothalamus, and that is the body's thermostat. It monitors the temperature of the blood. And we discussed that cutaneous vasodilation is when the blood vessels that are near the surface of your skin become dilated and engorged with blood when you're feeling too hot. Have you ever felt that if you're wearing a ring in the summer, it feels too tight? It's because the blood vessels dilate in the summer because it's so hot. And we're trying to shunt and remove the heat from the core, the viscera of our intestine and our stomach and our esophagus into the periphery, the outer superficial layers of the skin. Shivering thermogenesis, that's a spinal reflex that causes tiny alternating contractions of antagonistic muscle pairs. The contractions release heat from ATP consumption, and it can increase the body's heat production fourfold. Distribution of body heat in the hot and cold surroundings. Okay, disturbances of thermoregulation. We have fever. Fever is a normal protective mechanism that should be allowed to run its course if it is not excessively high. Disturbances of thermoregulation. So exposure to excessive heat can disrupt thermoregulation and result in heat cramps, heat exhaustion, and heat stroke. So exposure to excessive cold can disrupt the thermoregulation, and hyperthermia can cause life-threatening positive feedback loops. Alcoholism and drinking the alcohol. We discussed that alcoholism causes increased oxidative stress. Oxidative stress, where there will be a lot of ROSs, reactive, oxidative species. Alcohol metabolism is detoxified by hepatic and